Myself, Sami from tutor from Department of Biochemistry, Sri Lakshmi Narayana, Institute of Medical Science, Puducherry. And the topic is urea cycle and associated disorders. First, the ammonia will get released mostly in vertebrates as urea, in aquatic vertebrates, in terrestrial vertebrates as urea, and aquatic vertebrates as ammonium ion, and birds and reptiles it get excreted as uric acid. So the other source of ammonia is a toxic substance. It has to get released from our body. So the other source of ammonia are L-amino acid oxidase, glutaminase, asparginase, intestinal bacteria metabolism, and pyrimidine catabolism. So the ammonia produced by enteric bacteria will released in portal vein to the liver, and the ammonia produced by tissue by glutamine will released in liver, and in liver it get converted into urea, which is non-toxic and highly water soluble, whereas ammonia is a toxic substance, so it cannot be released by the kidney. So after formation of urea, the non-toxic substance is converted and then it is excreted mainly through kidney in urine. So the urea biosynthesis will occurs in 4 stages. First stage is transaminase, second is oxidative deamination of glutamate, third one is ammonia transport, and a reaction of urea cycles. So the nitrogen removal of ammonia acids. The alpha ammonia acid will get converted into keto acid in the transamination reaction, where alpha ketoglutarate is converted into glutamate with the help of PLP, where ammonia transferase. And then this glutamate goes under oxidative deamination to form ammonia. This ammonia combines with carbon dioxide to form urea, which is a non-toxic substance in the urea cycle. So glutamine is the main transporter form of ammonia between this tissue. So the glutamate is converted into glutamine with glutamine synthetase. So the ammonia production and the urea synthesis. The ammonia is produced in muscles, from muscles and from gut, which is protein intake, bacterial overgrowth, and from kidney, and it goes to the liver, and the liver it will produce urea, means urea synthesis will be taking place, and then the urea get transported to kidney, and then the urea is excreted in the urine. So the normal plasma level is 10 to 20 microgram per deciliter, where ammonia is a toxic. So the symptom of toxicity is tremors, slurring of speech, blurring of vision, coma, and finally the patient goes to the death. So the urea cycle or Krebs enzyme cycle, which incorporate nitrogen into urea, thus the prevention of accumulation of toxic nitrogenous compound. So the de novo synthesis of arginine will be taking place, and the site of synthesis is liver within the cell, mitochondria and cytosol. In mitochondria, there will be 2 reactions will be taking place. In cytosol, 3 reactions will be taking place. In mitochondria, 2 reactions are: in reaction 1, carbamyl synthase 1 and ornithine transcarbamylase. In cytosol, reaction 3, arginosuccinate synthase, reaction 4, arginosuccinate lyase, reaction 5, arginase. So the source of atom of urea, the carbon dioxide as bicarbonate, glutamine as NH3, aspartate as NH2. So this is the overview of urea cycle, where bicarbonate, ammonia, and 2-ATP with the carbamyl phosphate synthase 1, it converted into carbamyl phosphate, where ornithine and carbamyl phosphate combines to form citrulline, and this citrulline get out of the mitochondrial wall and it get entered into the cytosol, and this cytosol in the cytosol the citrulline combines with aspartate to form arginosuccinate, and here with the help of arginosuccinate synthase and the organosuccinate get divided into, or it get cleaved into arginine and fumarate with the help of organosuccinate lyase. This arginine, again arginine with the help of arginase, it divided into urea and ornithine. This ornithine will goes back to the mitochondria for another, for the formation of citrulline. And let's see one by one. The reaction one, the carbon dioxide plus ammonia with the help of 2 magnesium, so 2 ATP with the help of N-acetyl glutamate in the presence of carbonyl phosphate synthase 1, it converts into 2 ADP, and it finally it forms carbonyl phosphate. This reaction proceeds step by steps. The reaction of bicarbonate with ATP forms carbonyl phosphate and ADP. So the ammonia then displaced ADP, forming carbamate and orthophosphate. The phosphorylation of carbamate by second ATP then forms a carbamate phosphate. So the formation of carbamyl phosphate is, let's see here. Let's see here, this ammonia, the ammonia and carbon dioxide with the help of N-acetyl glutamate and carbamyl phosphate synthase 1, it forms carbamyl phosphate. Next, ornithine transcarbamylase is the enzyme where ornithine get converted into citrulline. The citrulline is transported out of the mitochondria through citrulline transporter in the inner membrane of the mitochondria. So next, it forms citrulline is converted into organosuccinate with the help of organosuccinate synthase, where citrulline get combined with aspartate to form organosuccinate with the help of organosuccinate synthase. So phosphate is an inhibitor of organosuccinate synthase, where organosuccinate again it get cleaved into arginine and fumarate with the help of organosuccinate lyase. So this arginine get cleaved to form urea and ornithine with the help of arginase. Ornithine and lysine inhibits arginase by competing with arginine. So ornithine is a transporter into mitochondria through ornithine transporter in the inner membrane of the mitochondria. So this is the overview of the urea cycle, where carbon dioxide enters into the mitochondrial wall and ammonia enter into the mitochondrial wall, where the reaction takes place to form carbonyl phosphate with the help of carbonyl phosphate synthase 1 and as well as N-acetyl glutamate. This carbonyl phosphate combines with ornithine, which is present in the mitochondria. It forms citrulline. Citrulline get transported out of the mitochondria with the help of citrulline transporter, and this citrulline get combines with aspartate to form organosuccinate with the help of organosuccinate synthase. This organosuccinate get cleaved to form fumarate with the help of organosuccinate lyase and arginine. This arginine get converted into ornithine and urea with the help of arginase, and finally this urea goes to the kidney, and then finally it is excreted through urine in the human body. And this ornithine get back into the mitochondrial wall, and it goes for the other reaction. So carbonyl phosphate synthase 1 is a pacemaker of enzyme of urea cycle. The activity of carbonyl phosphate synthase 1 is determined by N-acetyl glutamate, which is allosteric activator. So the concentration of the urea cycle enzyme can increases to 10 to 20 fold depending upon the ammonia load. For example, starvation elevated the enzyme levels were presumably to cope with increased production of ammonia that enhance the starvation induced degradation of the protein. Next, the regulation of urea cycle. The regulation of urea cycle will be taking place in two levels, where dietary changes causes induction and repression of urea cycle enzymes by 10 to 20 fold, where dietary intake of primarily protein, will increase the urea formation. Prolonged starvation also go for breakdown of muscle protein to form urea formation. So the rate of synthesis of carbamyl phosphate synthase 1 in the liver, and hormonal effects are corticosteroids, glucagon, growth hormone, catecholamines affects the level or activity of the enzyme, which also have the effect of gluconeogenesis. NAD on carbamyl phosphate synthase 1. N-acetyl-glutamic acid is a allosteric activator of carbamyl phosphate synthase 1. In high concentration of arginine, it stimulates the N-acylation of glutamate by acetyl-CoA. So the deficiency of the urea cycle enzymes are infant born with total deficiency of one or more enzymes have shortened the lifespan. Partially enzyme deficiency have altered Km value. Here all are aromatic amino acid defect except ornithine transcarbamylase, which is X-linked recessive. So intermediate can diffuse from hepatocytes, which get converted into accumulate in the blood and it get passed in the urine. If symptoms are not detected early, the severe mental retardation will be there, and then irreversible brain damage will be there by the ammonia. So the disorder associated with urea synthesis is, first one is N-acetyl-glutamate synthase deficiency, hyperammonemia type 1, hyperammonemia type 2, citrullemia, citrullemia type 2, citrullemia type 1 and citrullemia type 2, arginosuccinic acidemia and hyperarginemia. So one by one, and we will see the clinical features also with the enzyme defect. First one is MAG synthesis enzyme defect, which is N-acetyl-glutamate synthase, which causes main features encephalopathy, and hyperammonia type 1, which is aromatic amino acid defect, which has CPS1 enzyme defect, which is carbamyl phosphate synthase 1, which the clinical features are lethargy, hypothermia and apnea. Hyperammonemia type 2, which is X-linked ornithine transcarbamylase, is an enzyme defect, where lethargy, hyperammonia and apnea is a clinical feature. Citrullemia, which is aromatic amino acid defect, which is the enzyme defect is arginosuccinate synthase, where the clinical features lethargy, hypothermia and apnea will be there. The citrullemia type 2, which is aromatic amino acid defect, which is citrulline transporter is a main enzyme defect here. Main defect is citrulline transporter, means from mitochondria the citrulline transporter will not be take place from mitochondria to cytosol. So the main clinical features will be cholestatic jaundice, hepatic steatosis and psychiatric symptoms. And arginosuccinate acidemia, which is the enzyme defect is arginosuccinate lyase. This is trichosis. And hyperarginemia, which has enzyme defect of arginase, and triplet syndrome, which has enzyme defect of ornithine transporter, which the clinical feature is mentally retarded, seizures and pyramidal signs. So ornithine premise, which is ornithine transporter defect, which is mutation of ORNT1 gene from the ornithine transporter. Hyperornithemia, hyperammonemia and homocitrulluria syndrome, triplet syndrome. So mitochondrial carbamyl phosphate carbamates lysine to form homocitrulline. And thank you.